not change the sequence before or after it, right? So one of the amino acids is made wrong, but a complete protein is still made. Right?
Wait, so what do I put after having a point mutation?
It has a point mutation that changes the codon for that amino acid, right? So I get the wrong amino acid made within the sequence, but it does not change the sequence before or the sequence after, right?
How do you spell this? So you have a wrong amino acid that makes the right protein, or
It makes the wrong one.
okay, that's why I said one amino acid is off, making a wrong protein.
Yes. And
But
so,
it does not change.
but it does not change the sequence before or the sequence after. It only changes that one amino acid. But that change in that one amino acid gives you a slightly different protein, right? So if that protein is slightly different, then it's still not the right protein that's supposed to be made, right? So it's missense. I'm creating a protein, but I'm not creating the correct protein because of the change in that one amino acid. Correct? Now, nonsense. Nonsense one is where you have a point mutation in the DNA, right? That's going to create a codon that codes for what?
I thought, is it one nucleobase is wrong and a stop codon is made?
A stop codon is made.
Wait, for a stop codon?
Stop codon.
But before that,
Okay, create
point
codon.
You have a point mutation,
yeah.
right? And in that point mutation, it's going to create a stop codon. If I create a stop codon, am I going to be able to make a protein?
No.
No. Protein synthesis will end at that point. So if I don't produce the protein, is that going to be bad for the cell?
Mm-hmm.
Yes. So those are the three types of point mutations that we're talking about.
I don't even know what you're asking for because what is this?
What is what?
You put, like, on the one that was right there, you put silent, nonsense, and frame shift. But over here, you're talking about silent, missense, nonsense.
Because it got cut off when you, mine did the same thing. If you,
That's the one I got.
yeah.
But the things are silent, missense, nonsense, and the
Does anybody have one that has it not cut off? See, it was cut off.
Silent, missense, nonsense,
Yeah.
frame shift, insertion.
Okay, I'll remember that.
Just write it in. Okay, so now that brings us to a frame shift mutation, right? A frame shift mutation can occur with an insertion or a deletion of a nucleotide. Nucleotide.
Deletion
An insertion or a deletion of one of the nucleotides. So, say for instance, I have on my template strand, it is A, right? I've got 5 A's there. And I have an insertion of a T between the last A and the second to last A, okay? Now I have A, T, A, right? At that point, if I'm reading it this way, normally, that frame has just been shifted that way. And so now, that T that got put in the middle there, now is going to be reading wrong, right? So I got the right amino acid, and then all of a sudden I got the wrong amino acid, but then that changes the way everything else is read after that point. So once that is wrong, those 3 that get read are going to be wrong. Those 3 that get read are going to be wrong. So everything is wrong after that point. So that changes the whole way that the frame has been read. That's why they call it a frame shift. Okay? I'm still gonna produce a protein, but it's going to be a major difference in the protein. Okay? Now, if I was looking at, I don't know if you've seen, okay, let's talk about a train, right? You have your engine of the train, right? And you have the second engine of that train, and then you have your first car, right? Normally, I'm going to have a bunch of coal cars right after that one, right? Because that's the way it's programmed. But because there is an addition to my information, an insertion of a nucleotide, that changes the whole plan. So instead of a bunch of coal cars, now I have a bunch of cattle cars. Changes the whole thing. It's a big difference between cattle cars and coal cars. You got that?
Yeah, I got
Okay, now, same thing happens with a deletion. What a deletion is, I have a deleting of a nucleotide. Changes the whole way the frame is made. So,
Deleting of a nucleotide?
deletion, yes. So if I take one away, rather than A, 5 A's and then a G, if I take away one of those A's, now I have A, G. Instead of that A there, it changes the whole way that it's read. So now it's a completely different protein. So you have a major difference in the type of protein made.
So if it's already AAG, and you take one of the G's out,
Uh-huh,
that's deleting?
that's a deletion, yes.
Oh, okay. I get it.
And so it causes the same kind of mutation, such as a major change in the protein that's made. So everybody got that? Okay.
Either insertion or deletion. Sorry.
A deletion,
Adding or subtracting.
adding or subtracting will cause the same result.
Okay,
A major change in the protein that's made.
okay, major
I
change.
get just one.
Yes.
This one, I did because,
Okay, let's look at the last three here. Explain the horizontal gene transfer through transformation by explaining Griffith's experiment, okay? We did this in the lab. We did a transformation in the lab. We saw the results, right? So the whole basis of transformation is the fact that bacteria can take up DNA in their environment. Sometimes we call this naked DNA because it's not really encapsulated in there, okay? So it can take up DNA in its environment and incorporate it into its own genome and take on those traits, okay? What Griffiths did is he had two types of bacteria. He had a capsulated bacteria, the pneumococcal bacterium, right? It was pathogenic. He injected that into a mouse. What happened to the mouse? It died because it had the capsulated virulent bacteria. He then took that virulent bacteria and he heat killed it. He heated it up so that he killed it, right? He then injected that bacteria into a mouse. What happened to the mouse? It lived. He had another form of that bacteria, but this one did not have a capsule around it. So it was not virulent. He injected that into a mouse. What happened to the mouse? It lived. Then he took the heat-killed bacteria and he put it into the same test tube as the non-virulent, non-capsulated bacteria, and he incubated them, okay? What happened during this incubation?
no, when that was heat fixed, like it.
It gave off its DNA. If it gave off its DNA in that environment, what happened to the non-virulent bacteria?
It took it in and
It took it
changed.
in. Okay, once it took it in, what was it? What was the reaction of that? What aftermath? Well, we're not talking
It
about the
became
mouse yet.
pathogenic.
Huh?
It's transformed into a live.
It transformed into a live version of the pathogenic one because it took up that DNA, incorporated it into its genome, and now it is now expressing the virulent form of that bacteria. So he injected that into the mouse. What happened to the mouse? He died. When he took a sample from that mouse and he looked at it under the microscope, what did he see?
Not the non-pathogenic became pathogenic.
He saw a pathogenic bacteria. The reason why he saw a pathogenic bacteria is because that non-pathogenic bacteria took up the DNA from the killed one, incorporated it, started expressing virulence. Okay?
So he's had seen that a non-pathogenic became pathogenic.
Yes, a non-pathogenic became a pathogenic because they like to share DNA.
So, the moral of the story.
Don't hang out with pathogenic bacteria, dead or alive.
Okay.
Okay, so we can put that in there, his moral of the story.
What's Vera?
Virulence.
What?
Virulence, meaning the ability to cause disease.
So, but we can just put Vera?
Virulence?
You were not saying Vera?
I didn't say virum. Why would I say virum?
I swear that's what you said.
No.
You talked.
I may have said bacterium.
No, you said Vera.
I never said
I
virum.
swear you said.
No, negative.
I heard it like 3 times.
Who heard me
So-
say virum?
I don't know.
Nobody.
I don't,
Look, you
Okay,
listen to
well, you
it.
got
He's
it
gonna
before.
say
You can
it.
check the video out
So
later.
exactly what are you looking for on the answer? We need to state
Like
what
YouTube.
horizontal gene transfer is,
Like I
and
posted
Okay.
then tell about the mouse
What is
experiment.
horizontal gene transfer?
But I'm gonna
It's
do
where
it.
naked DNA
Like,
is
it's gonna be
uptake by
like-
conjugate.
Okay, no,
Two.
no. The
No?
definition of horizontal
Better to tell
gene
'cause it's in the
transfer
cafe.
is transferring
I just
of
put it down
genetic
into
material from one bacterium to
microbiology
another
exam
bacterium.
3.
There are 3 different
Part one.
ways this can occur.
Yeah,
Trans-
my
You
username
can't
is Jimmy
guess.
Torres.
Yeah.
I have your.
Here, you just said urea.
What?
Yeah,
See, you said
and it should
yeah.
be posted here for it.
Or bacteria.
Like right now it's the classroom one. It takes a bit
Your
to put
recording
it
is
on there.
wrong.
Yeah.
Anyways, transferring. We're transferring
One bacteria or another bacteria.
we're transferring DNA or genetic material from one bacterium to the other.
Okay.
That's the definition of horizontal transfer. Okay. Yes. Now, next one is explain in detail how generalized transduction occurs. Transduction involves what?
Can you draw this chart?
What's that chart?
If you like label it.
A transduction?
Help us.
Yeah.
Sure.
Where do you get that? That wasn't very convincing.
But he won't give you points for it.
Well, no, but it'll help me remember.
I put a
the
smiley
answers.
face next to the drawing if it's really good.
Because this is what you had on the board, so.
I put a smiley face next to the drawing if it's really good. Whatever helps you write it, do it.
But remember, you only have 50 minutes.
You show up to class
It
early.
takes you all 2 minutes to draw that.
Yeah, I was gonna say, I don't think this takes very long.
No.
It's just-
Anyways,
It's really good.
generalized transduction involves what?
Where a phage or
Phage,
virus
phage.
injects DNA into the bacteria, causing the bacteria DNA to synthesize and make copies of the virus DNA and host DNA and is injected into the donor DNA and incorporated into the donor's chromosomes.
Wait, what's
Okay.
the third
Can you repeat that?
So,
Injects DNA?
what's going on is a virus that is specific for bacteria. We call that a bacteriophage, right? When the phage infects the first donor, right? When it's released, it picks up a piece of DNA from that donor with it. When it goes to infect a recipient, when it injects its DNA into that recipient, part of that donor DNA has been trapped into the phage. So it will then go into the donor, into the recipient. The recipient will then take that up into its genome, incorporate it, and express those traits. So how does this happen? You have a bacteria minding his own business. A bacteriophage will then land on it and inject its DNA into